receptors, GPCRs, are essential in physiological processes and play a significant role in students with a comprehensive understanding of GPCR. GPCRs are a diverse family of cell surface receptors that transmit extracellular signals into intracellular responses. They are involved in the regulation of numerous physiological processes and serve as important drug targets. The structure of GPCRs consists of 7 transmembrane helices, with the N-terminus located extracellularly and the C-terminus intracellularly. Ligand binding to GPCRs triggers conformational changes, activating downstream signaling pathways. Ligands for GPCRs can be a diverse range of molecules, such as neurotransmitters, hormones, peptides, and small molecules. These ligands specifically bind to the extracellular domain of GPCRs, inducing a conformational change in the receptor. Upon ligand binding, GPCRs interact with heterotrimeric G proteins, which are composed of alpha, beta, and gamma subunits. This interaction leads to the activation of the G protein and subsequent downstream signaling events. In this section, we will delve into the pathways and responses that occur within cells when G protein-coupled receptors, GPCRs, are activated. Activation leads to intracellular signaling events, involving the exchange of GDP for GTP on the G-alpha subunit, followed by dissociation of the G-alpha subunit from the G-beta-gamma subunit. The liberated G-alpha subunit then interacts with different effector molecules like adenylyl cyclase, phospholipase C, or ion channels. This interaction influences the levels of various intracellular second messengers, including CAMP, IP3, and calcium ions. These second messengers are vital in triggering specific cellular responses. To maintain cellular homeostasis and prevent excessive signaling, GPCR activity is tightly regulated. One crucial regulatory mechanism is desensitization, which involves the uncoupling of the receptor from the G protein upon prolonged exposure to the ligand. Desensitization can occur through several mechanisms, including receptor phosphorylation by GPCR kinases, GRUKs, and subsequent recruitment of beta-arrestins. This leads to the internalization of the receptor via endocytosis, preventing further signaling. Receptor phosphorylation by GRUKs plays a crucial role in GPCR signaling. Phosphorylated receptors have a higher affinity for beta-arrestins, which further modulate downstream signaling pathways. Beta-arrestins not only desensitize GPCR signaling but also serve as scaffolds for the recruitment of signaling molecules, such as kinases and adapters. This allows the formation of signalosomes, which mediate diverse cellular responses. Aside from G proteins and beta-arrestins, GPCRs interact with a myriad of other intracellular proteins, such as GPCR-associated sorting proteins, GASPs, receptor activity modifying proteins, RAMPs, and PDZ domain-containing proteins. These interactions fine-tune GPCR signaling and can modulate receptor trafficking, dimerization, and signaling specificity. Understanding these protein-protein interactions is crucial for unraveling the complexity of GPCR-mediated cellular responses. Membrane lipids, particularly cholesterol and phospholipids, play a vital role in GPCR regulation. They contribute to the structural organization of the membrane and can directly interact with GPCRs, influencing their activity and localization. Cholesterol, for instance, affects GPCR clustering and stability in lipid rafts. Phospholipids, on the other hand, can regulate GPCR function by modulating receptor conformation and G protein coupling. The lipid composition of the membrane, therefore, impacts GPCR signaling. Bias signaling refers to the selective activation of specific downstream signaling pathways by GPCRs, even in the presence of multiple ligands. This phenomenon highlights the complexity and versatility of GPCR signaling. Various mechanisms contribute to bias signaling, including ligand-specific conformational changes in the receptor, biased ligand binding kinetics, and differential activation of intracellular signaling effectors. Understanding bias signaling can aid the development of selective drugs targeting specific signaling pathways. GPCRs play crucial roles in both normal development and disease processes. They are involved in organogenesis, neuronal migration, angiogenesis, and immune cell development. Furthermore, dysregulation of GPCR signaling is implicated in various diseases, including cancers, cardiovascular disorders, neurological disorders, and metabolic syndromes. Elucidating the specific roles of GPCRs in these disease processes offers potential therapeutic targets. Summarize in 33 words: GPCRs play a crucial role in sensory perception. They detect odorant molecules, allowing us to differentiate smells. They also recognize taste molecules, enabling taste perception. In the visual system, GPCRs initiate the cascade for vision. Studying GPCR function can help understand sensory disorders and develop therapies. GPCRs play a crucial role in regulating immune responses and inflammation. They are involved in leukocyte migration, chemotaxis, cytokine production, and immune cell activation. Inflammatory processes rely on GPCR signaling to coordinate immune responses. Dysregulation of GPCR-mediated immune signaling can lead to immune disorders, autoimmune diseases, and chronic inflammatory conditions. Targeting GPCRs in immune regulation holds promise for therapeutic intervention. Scientists studying G-protein-coupled receptors, GPCRs, use various methods to study their function and develop drugs. These include studying receptor structure, screening for binding compounds, and using animal models. New tools like CRISPR-Cas9 gene editing help in understanding GPCR genes, and crystal structures aid in designing targeted drugs for improved therapies. In conclusion, G-protein-coupled receptors, GPCRs, are vital in cellular signaling and have profound implications for disease and drug discovery. Understanding their biology, function, and potential therapeutic applications is essential for medical students.